Welcome to the Wonder Lab. Today, we dive deep into the science of love, desire, and attachment, exploring how our brains, our bodies, and even subtle interactions shape the way we connect with others. From the hidden chemistry that drives attraction to the neurobiology behind longing and loyalty, we'll uncover the fascinating mechanisms that influence how we fall in love, why we stay, and what makes desire thrive. This is not just about emotions. It's about the science that governs them. Asterisk, in the beginning of every connection, something remarkable occurs. Two individuals, strangers at first, begin a dance that is both subtle and profound. It is a dance guided not just by conscious thought, but by biological mechanisms that operate beneath our awareness. These mechanisms influence the way we perceive each other, the way we experience desire, and ultimately, the way attachment forms. Science tells us that love is more than just a feeling. It is a system, an orchestra of neurochemicals, hormones, and brain circuits, all interacting in precise ways. Dopamine, the neurotransmitter of motivation, sparks the thrill of attraction. Oxytocin, often called the bonding hormone, strengthens the ties that form between two people, encouraging trust and intimacy. And yet, this dance is not only chemical, it is also narrative. The way we interpret our partner's words, gestures, and intentions shapes the way our autonomic nervous system responds. Consider the concept of scent, a principle studied extensively in psychology. Some individuals experience profound growth and pleasure when their partners act in ways that make them feel admired, understood, or inspired. These gestures do not need to be grand. They can be as subtle as a thoughtful word, a shared experience, or a sign of recognition that the other person truly values them. When self-expansion occurs, the brain releases chemicals that elevate mood, enhance self-perception, and, importantly, influence the way potential alternative partners are perceived. The feeling of being filled up by connection makes other attractions seem less potent, creating a stabilizing effect on the relationship itself. But attachment is not solely a function of self-expansion. Our bodies carry signals of compatibility and attraction that we barely notice. Pheromones, subtle odors, and even the rhythm of another person's heartbeat can communicate levels of genetic fitness, compatibility, and emotional resonance. Studies have shown that hormonal cycles, both in men and women, affect perceptions of attractiveness and desire. Birth control, for example, can subtly shift these perceptions, demonstrating that attraction is as much biological as it is psychological. From the earliest stages of romance, desire is intertwined with prediction. Our brains constantly evaluate: is this person trustworthy? Are they invested? Do they care about me? Each gesture, each expression, each word of affirmation adds to a mental modeler, continuously updated map of emotional and physical reward. The more this model confirms safety, excitement, and value, the more desire and attachment strengthen. Conversely, when signals are inconsistent or absent, the nervous system can sense imbalance, increasing attention to other potential partners and creating vulnerability. Yet, even within this complex interplay, the conscious mind plays a role. We may choose how we act, how we respond, and how we cultivate connection. Awareness of the mechanisms at work, the neurochemistry, the self-expansion, the hormones gives us the opportunity to engage more intentionally, to craft experiences that not only spark attraction but deepen attachment, to use knowledge of the brain and body not as a tool for manipulation, but as a guide to understanding the profound mystery that is human connection. And so, in the first moments of attachment, we see the convergence of biology, psychology, and choice. The chemical signals of desire blend with the subjective experiences of meaning and value. The autonomic nervous system, the limbic system, and the prefrontal cortex work in concert, shaping the trajectory of our relationships. It is in these earliest interactions, often unnoticed, that the seeds of love, attachment, and enduring desire are planted. As relationships deepen, something extraordinary unfolds within the human brain. It is not merely that two people spend time together or share experiences, it is that their presence fundamentally reshapes each other's perception of the world, themselves, and even others outside the relationship. Scientists describe this as some people are particularly sensitive to it. The way their partner's words, gestures, and behaviors can make them feel more competent, more valued, more alive. A simple compliment, a shared adventure, or the subtle acknowledgement that one is indispensable in the relationship triggers profound neurochemical responses. Dopamine surges, serotonin levels adjust, oxytocin binds to receptors, and the autonomic nervous system shifts to a state of reward and engagement. Interestingly, studies have shown that self-expansion influences not only feelings toward one's partner, but also toward the outside world. When individuals are primed to experience self-expansion, when they hear narratives from their partner highlighting excitement, novelty, or admiration, they rate alternative potential partners as less attractive. It is as if the brain, filled with the reward of connection, no longer needs to search for stimulation elsewhere. This is not a conscious choice. It is a neurobiological reality. Yet, not all individuals respond equally. Some are more dependent on this partner-driven self. Without it, they are more likely to notice the attractiveness of others. Their internal state is like an unfilled cup, until their sense of value and excitement is replenished by the partner. They remain sensitive to external stimuli. This reveals a critical insight. The stability of attachment is deeply connected to the ability to regulate one's own self-worth internally, and the extent to which one relies on the partner for emotional and autonomic fulfillment beyond self-expansion. The neurochemistry of desire itself is intricate. Libido is not dictated solely by testosterone or estrogen. It is a coordinated dance of hormones, neurotransmitters, and autonomic states. Testosterone and its derivatives, such as dihydrotestosterone, enhance the drive to seek mates, while estrogen similarly influences desire in both sexes. The balance between these hormones, coupled with dopamine signaling, shapes not only the intensity of sexual desire, but also the ability to act upon it. This is why increasing dopamine alone does not necessarily enhance libido. Too much dopamine can hyperactivate the sympathetic nervous system, driving individuals into states of arousal that make pursuit possible but engagement difficult. The parasympathetic system, responsible for sexual readiness and physical response, must remain accessible. It is a delicate seesaw. Arousal must oscillate between excitement and readiness. Misbalance, whether through drugs, excessive stimulation, or hormonal manipulation, can lead to chronic desire without the ability to fulfill it, a phenomenon observed in both recreational drug users and individuals on certain medications. Supplements, however, can provide subtle yet effective modulation. 3. In particular, maca root, tonkat oli, and Tribulus terrestris have been shown to influence libido through different pathways. Maca, consumed in doses of 2 to 3 grams per day, increases subjective sexual desire without significantly altering testosterone or estrogen. Tongkat Ali, particularly the Indonesian variety, can increase free testosterone by reducing sex hormone binding globulin, thereby enhancing sexual function and drive. Tribulus terrestris shows promising effects, particularly in women, by increasing bioavailable testosterone and improving sexual function in populations with diminished libido. But beyond these supplements, the most fascinating element remains the subconscious processes that drive attraction. Odors, pheromones, taste, even minute rhythms in movement, all communicate compatibility and desire in ways that bypass conscious awareness. Humans are wired to respond to these subtle cues. One study noted that women's attraction to men varied with hormonal cycles, while men's perception of women's attractiveness also shifted in response to chemical and olfactory signals. Even birth control can modulate these unconscious preferences, underscoring the complex interplay of biology and behavior. This interplay also shapes the narratives we construct about our partners and ourselves. The stories we tell, whether internally or shared with each other, affect autonomic arousal, desire, and attachment. When a partner describes the relationship as exciting, challenging, and growth-inducing, the listener's brain is flooded with signals of reward, significance, and emotional safety. This self-expansion strengthens the bond, making alternative partners seem less compelling. Conversely, without such reinforcement, the same neurochemical landscape leaves the individual more open to external attractions. In essence, the first months and years of attachment reveal a dual reality. Love and desire are both biological and psychological, conscious and subconscious. They are shaped by hormones, neurochemicals, gestures, narratives, and deeply ingrained subconscious processes. And in understanding these processes, we begin to glimpse the profound mechanisms that allow humans to form lasting connections, navigate attraction, and cultivate stable relationships that endure across time. As we continue exploring the biology of love, desire, and attachment, it becomes clear that human sexuality and attraction are not simple, linear processes. They are multifaceted, influenced by hormones, neurotransmitters, and the intricate orchestration of the autonomic nervous system. Let's begin with testosterone and estrogen, two hormones often cited in the context of libido. Testosterone, often simplified as the male hormone, plays a critical role in sexual motivation and pursuit in both men and women. Its effects are nuanced, unbound, or free. Testosterone interacts with receptor sites in the brain and body to drive sexual desire, enhance mood, and promote exploratory behavior toward potential mates. But testosterone does not act in isolation. Estrogen, frequently overlooked in discussions about libido, is equally essential. In women, estrogen levels fluctuate across the menstrual cycle, affecting attraction, receptivity, and desire. In men, estrogen, derived from the aromatization of testosterone, modulates libido, sensitivity, and even emotional connection during sexual activity. Together, these hormones create a delicate hormonal dance, guiding sexual behavior and desire. The ratio between testosterone and estrogen is critical. Too much or too little of either hormone can disrupt desire, not just by changing libido, but by altering the way the brain perceives reward, motivation, and connection. This is why hormonal supplementation or therapies, when done incorrectly, can be counterproductive, increasing one hormone without consideration of the whole system may amplify sexual desire but hinder actual engagement. Neurotransmitters are equally important in this landscape. Dopamine, the brain's chemical of motivation, reward, and pursuit, plays a central role in desire. Dopamine surges when anticipating reward, including sexual reward, and drives the motivation to pursue partners. But dopamine is part of a larger system. It interacts with serotonin, norepinephrine, and endorphins, creating the emotional and physical states we recognize as arousal, excitement, and attraction. This interaction explains why simply trying to increase dopamine through supplements, drugs, or behavioral hacks does not automatically increase sexual performance or satisfaction. High dopamine can induce a state of constant pursuit or hyperarousal. But if the parasympathetic system is not engaged, the body cannot translate desire into physical readiness or enjoyment. The result is a disconnect between motivation and action, a phenomenon observed in individuals using stimulants, certain antidepressants, or other pharmacological agents beyond hormones and neurotransmitters. The autonomic nervous system shapes sexual experience. The sympathetic system primes the body for pursuit, energizing the body, heightening attention, and preparing for action. The parasympathetic system allows for engagement, relaxation, and physical response. Optimal sexual function requires a dynamic balance between these two arms, a seesaw of arousal, where the body is excited but not overactivated, primed for engagement but not frozen in pursuit. Misalignment in this system explains why stress, excessive stimulation, or certain medications can reduce libido. Despite the presence of desire, supplements offer one avenue for modulating these systems safely. Maca root, Tonkat oli, and Tribulus terrestris have been studied extensively for their effects on libido and sexual function. Maca, for instance, appears to increase sexual desire in both men and women without altering testosterone or estrogen levels significantly. Tonkat oli, particularly the Indonesian variety, may raise free testosterone levels by reducing sex hormone-binding globulin, enhancing libido and sexual function. Tribulus, studied primarily in women and older populations, shows potential to increase bioavailable testosterone and improves sexual function, although its effects can vary based on individual physiology. Importantly, these supplements act in conjunction with the body's intrinsic neurochemical systems. They do not replace the need for self-expansion, partner interaction, and emotional connection. In fact, their effectiveness is often amplified when individuals are already experiencing positive relational dynamics. The praise, support, and stimulation that come from meaningful attachment and mutual admiration. Supplements may enhance libido, but self-expansion, the feeling that one is growing, valued, and engaged within a relationship drives sustained desire and attraction. Subconscious processes further shape attraction and desire. Humans are sensitive to pheromones, chemical signals, and olfactory cues that influence mate selection unconsciously. Studies reveal that women's attraction to men shifts across their menstrual cycles, influenced by subtle hormonal signals. Men's perception of female attractiveness is also modulated by chemical cues, including scent and pheromones. These signals operate beneath conscious awareness, guiding behavior, desire, and emotional connection. Even birth control can alter these preferences, highlighting the profound impact of biology on human relationships. Yet, desire and attachment are not solely dictated by biology. The narratives we construct about our partner, ourselves, and the relationship profoundly influence the neurochemical landscape. Hearing a partner describe the relationship as exciting, challenging, and growth-oriented triggers self-expansion, enhancing dopamine, oxytocin, and reward signaling. This, in turn, reduces the perceived attractiveness of potential alternative partners and strengthens commitment. Conversely, when self-expansion is absent, the same biological systems may heighten sensitivity to external stimuli, increasing perceived attractiveness of others. Attachment, therefore, is both biological and psychological. Secure attachment emerges when individuals maintain stable internal representations of self, can regulate autonomic and emotional states, and experience growth and validation within the relationship. Insecure attachment arises when self-expansion is tied too rigidly to a partner, making individuals vulnerable to external temptations and fluctuations in perception. This interplay highlights the importance of balancing internal self-worth with partner-driven reinforcement. A stable internal state supports long-term attachment, while external validation enhances but should not define. Self-expansion. Ultimately, desire, love, and attachment are emergent phenomena of complex neurobiology. Hormones, neurotransmitters, autonomic states, and subconscious processes converge to create the human experience of attraction. Self-expansion amplifies these effects, influencing perceptions of others and reinforcing bonds. Supplements may support libido, but the foundation remains in neurochemistry, connection, and internal regulation. In this light, love is not a simple choice, nor is desire merely a feeling. It is a delicate orchestration of biology and psychology, of conscious thought and subconscious signaling, of hormones and neurotransmitters, of self and other. Understanding this system reveals the profound sophistication of human relationships and provides insight into the subtle, invisible forces that drive attraction, sexual desire, and the attachment bonds that define our lives. As we step deeper into the landscape of love, desire, and attachment, it becomes evident that knowledge alone is insufficient. Awareness of hormones, neurotransmitters, and autonomic states is crucial, but the application of this knowledge transforms human connection from a passive experience into an active, engaged journey. One of the most effective methods to strengthen attachment is through shared experiences that trigger self-expansion. Self-expansion is the process of growth, novelty, and learning that enriches the self while simultaneously enhancing relational bonds. Neuroscientific studies reveal that couples who engage in novel, challenging activities together, be it travel, learning a new skill, or even solving complex puzzles, show increased dopamine release, stronger reward system activation, and heightened feelings of closeness. This isn't mere anecdote. Functional MRI scans demonstrate that shared novel experiences activate the same reward circuits as sexual intimacy, creating a multi-layered network of reinforcement in the brain. Moreover, practical application of oxytocin release has profound effects on attachment and sexual satisfaction. Oxytocin, often called the bonding hormone, is released during physical touch, eye contact, shared laughter, and meaningful conversation. Regular, intentional engagement, holding hands, gentle caresses, and synchronized movement boosts oxytocin levels, enhancing trust, reducing stress, and reinforcing emotional attachment. Interestingly, oxytocin release is amplified when the brain perceives reciprocity and attunement, a partner's mirroring of emotion. Body language or tone strengthens the neurochemical connection and deepens relational satisfaction. Beyond oxytocin, deliberate modulation of dopamine and serotonin pathways can enrich sexual desire. Behavioral scientists recommend structured reward schedules in relationships. For instance, small gestures of appreciation, surprise compliments, or unexpected acts of kindness create intermittent dopamine spikes similar to the reward mechanisms in gambling or gaming that keep the brain engaged and motivated, simultaneously maintaining serotonin balance through gratitude exercises. Mindfulness and shared reflection enhances mood stability, reducing the irritability or emotional fatigue that often undermines long-term attachment. Hormonal optimization continues to play a supportive role. Testosterone and estrogen, when regulated naturally through exercise, sleep, and nutrition, provide a consistent substrate for desire. Resistance training, high-intensity interval workouts, and adequate protein intake enhance free testosterone levels, while phytoestrogen-rich foods and balanced micronutrients support optimal estrogen function. Sleep, often overlooked, is paramount. During slow-wave sleep, testosterone synthesis peaks, cortisol levels drop, and the brain consolidates emotional learning, all of which directly influence libido and emotional resilience. Psychological strategies also reinforce desire and attachment. Imagination and novelty are powerful tools, mentally envisioning scenarios of shared adventure or pleasure primes the brain's dopaminergic system, preparing the body and mind for heightened engagement. Couples who deliberately introduce novelty in communication, whether through playful teasing, creative storytelling, or unexpected surprises, activate the same reward circuits as real-life adventure, strengthening emotional and sexual bonds. Mindfulness, another critical component, enhances awareness of autonomic nervous system states. By observing bodily sensations without judgment, individuals can detect early signs of stress, fatigue, or overactivation, allowing intentional regulation of sympathetic and parasympathetic balance. Mindful touch, deep breathing, and guided attention during intimate moments not only optimize sexual performance but also reinforce emotional attunement. As both partners are fully present and synchronized, the neurochemistry of attachment is further influenced by gratitude and reflective practice. Writing or verbalizing appreciation for a partner enhances serotonin and oxytocin, fostering secure attachment. These practices are not trivial. Longitudinal studies show that couples who engage in structured gratitude exercises exhibit stronger relational satisfaction, fewer conflicts, and higher sexual fulfillment over time. Additionally, understanding pheromonal and olfactory cues can subtly enhance attraction. While these signals operate below conscious awareness, they guide mate preference and reproductive strategies. Fragrance choices, natural body scent, and even dietary influences can modulate attractiveness. Synchronizing these cues with deliberate behaviors that promote closeness, shared touch, eye contact, mutual laughter, amplifies both desire and attachment. Supplements can complement these practices. Adaptogens like rhodiola rosea and ashwagandha reduce stress, indirectly supporting sexual function by lowering cortisol, while L-citrulline and nitric oxide boosters enhance vascular function, improving arousal physiology. Yet, these are adjuncts. Without the behavioral, emotional, and psychological strategies in place, their impact is limited. Finally, the integration of all these systems, neurochemical, hormonal, autonomic, psychological, is what defines sustainable desire and attachment. Relationships are living systems requiring attention, feedback, and adaptation. The brain and body respond not only to direct stimuli, but also to patterns, rituals, and narrative frameworks. Couples who cultivate shared purpose, emotional attunement, and structured novelty activate their neurochemical reward systems repeatedly, creating resilience against the natural fading of passion over time. In essence, enhancing love and desire is an art grounded in science. It requires understanding the biological foundation hormones, neurotransmitters, autonomic regulation, while applying psychological and behavioral strategies, self-expansion, mindfulness, gratitude, and novelty. When combined, these approaches produce a rich tapestry of sustained attraction, deep attachment, and emotional fulfillment. Love, in this perspective, is not merely a feeling, but a dynamic, trainable system, shaped by intentional actions, awareness, and connection. As we journey further into the intricacies of love and attachment, it becomes clear that initial attraction and early desire, while powerful, are not sufficient to sustain long-term relationships. The science of attachment reveals that human bonds are not static, they are dynamic systems, continuously shaped by our neurobiology, behavior, and shared experiences. Central to this understanding is the concept of attachment styles, first described by John Bowlby and later expanded in adult relationships. Individuals with a secure attachment style experience comfort with intimacy, trust in their partner, and a healthy balance of independence and closeness. Neuroscientific studies show that secure attachment correlates with more stable autonomic nervous system activity, higher oxytocin and dopamine responsiveness, and reduced cortisol reactivity in stressful relational contexts. These physiological patterns support resilience, emotional regulation, and sustained desire. In contrast, anxious attachment manifests as hypervigilance to partner signals, heightened sensitivity to perceived rejection, and frequent cycles of worry. Brain imaging reveals increased amygdala activation in response to partner separation or conflict, coupled with fluctuations in reward circuitry responsiveness. While this attachment style can intensify desire in short bursts, it often undermines long-term stability, as the neurochemical system is repeatedly taxed by stress and uncertainty. Conversely, avoidant attachment is characterized by a preference for emotional distance, self-reliance, and reduced expression of need or vulnerability. Avoidantly attached individuals demonstrate dampened oxytocin release in relational contexts, reduced activity in social reward circuits, and a heightened capacity for self-soothing through independence. While this may protect against immediate relational stress, it can also diminish intimacy, sexual satisfaction, and the experience of deep emotional connection. Understanding these attachment patterns is not merely academic. It informs practical strategies to enhance desire, intimacy, and relational satisfaction. Securely attached individuals benefit most from consistent positive reinforcement, shared novelty, and emotional attachment, whereas anxious or avoidant individuals require structured interventions, conscious communication strategies, validation exercises, and regulated exposure to intimacy to retrain the brain's reward and stress pathways. Neurochemical systems provide a tangible framework for intervention. Oxytocin, as discussed earlier, is crucial for bonding, but its release is context-dependent. Physical touch, synchronized activity, and verbal affirmation trigger oxytocin release more effectively when paired with emotional attunement. Dopamine, associated with reward, motivation, and pursuit, interacts with oxytocin to reinforce attraction and attachment. However, as previous studies suggest, excessive dopamine without parasympathetic balance can lead to arousal without intimacy, a state of frustrated desire. Hormonal influences, particularly testosterone and estrogen, remain integral to desire throughout a relationship. In men, testosterone supports libido, sexual pursuit, and reward sensitivity. In women, estrogen modulates sexual receptivity, emotional sensitivity, and partner preference. Importantly, both hormones fluctuate naturally with age, stress, and lifestyle factors. Long-term relationship satisfaction often correlates with partners' awareness of these cycles and intentional adaptation. Whether through sexual timing, shared novelty, or emotional reinforcement, self-expansion continues to play a central role in sustaining passion. Couples who cultivate experiences that expand personal and relational horizons, learning new skills together, pursuing shared goals, or exploring unfamiliar environments activate dopaminergic and reward pathways in tandem with oxytocin release. This creates neurochemical synergy. Novelty drives motivation and excitement, while bonding hormones reinforce safety and attachment. Over time, these experiences buffer against the natural decline of desire that often accompanies routine and predictability. Practical neuroscience suggests structured novelty is particularly effective. The brain responds most robustly to stimuli that are challenging but attainable, engaging curiosity, reward anticipation, and mastery. For example, learning a musical instrument, practicing a new sport together, or tackling cooperative problem-solving tasks enhances relational satisfaction. Crucially, these experiences must be mutual. The self-expansion effect is amplified when both partners are invested, creating a feedback loop of reward, reinforcement, and attachment mindfulness, and emotional attunement remain powerful tools. Training awareness of autonomic states, such as noticing tension, elevated heart rate, or shallow breathing, allows intentional activation of the parasympathetic nervous system during intimate encounters. Practices like synchronized breathing, eye gazing, and gentle touch regulate sympathetic arousal, improving sexual responsiveness, emotional attunement, and relational resilience. Beyond behavior and neurochemistry, subconscious sensory cues influence desire. Pheromones, body scent, and even vocal tone operate below conscious awareness, shaping mate selection and sexual attraction. While modern humans often rely heavily on visual and cognitive cues, studies indicate that olfactory and tactile signals continue to exert measurable effects on partner preference, sexual arousal, and relational chemistry. Supplements may provide auxiliary support for desire and arousal. Compounds such as maca, tonkat, oli, and tribulus terrestris have demonstrated efficacy in enhancing libido across populations. Maca root, at doses of 1.5 to 3 grams per day, increases sexual desire even without altering testosterone or estrogen. Tonkat oli may increase free testosterone, enhancing libido and motivation. Tribulus has shown mixed but promising effects, particularly in females, for boosting desire and sexual function. These interventions complement behavioral and neurochemical strategies. Though they are not substitutes for the foundation of trust, novelty, and emotional attunement, long-term attachment also benefits from intentional ritualization. Daily practices of gratitude, appreciation, and acknowledgement of partner value reinforce reward pathways, elevate oxytocin, and mitigate conflict-related cortisol spikes. Studies demonstrate that couples engaging in structured gratitude exercises report higher relational satisfaction, reduced emotional volatility, and more robust sexual desire over time. Finally, love and attachment must be understood as dynamic, trainable systems. Relationships are living networks of neurochemical, hormonal, psychological, and behavioral processes. Initial attraction is only the beginning. Sustained desire and attachment arise from continuous engagement, novelty, emotional attunement, and mutual growth. The brain, autonomic nervous system, and hormonal milieu respond to consistent patterns of reinforcement, creating resilience, deepened connection, and long-term relational fulfillment. In conclusion, the maintenance of love and desire in human relationships requires an integrated approach. Awareness of attachment styles, intentional behavioral strategies, neurochemical modulation, hormonal optimization, and structured novelty. When applied with consistency and mutual engagement, these strategies transform the raw potential of attraction into a durable, resilient, and deeply satisfying partnership. As we delve deeper into the complexities of human attachment, one of the most challenging aspects of romantic relationships emerges: the management of infidelity and jealousy. While love and desire are often framed in idealized terms, the reality of long-term partnerships involves navigating threats to trust, competition, and the fragile balance of emotional security. Infidelity is not merely a moral failing, it is rooted in the interplay of evolutionary pressures, neurobiology, and relational dynamics. From an evolutionary perspective, humans evolved in contexts where sexual access, genetic diversity, and resource distribution influenced reproductive success. Both men and women are biologically wired to be sensitive to cues of partner fidelity, though these sensitivities manifest differently. Men, historically concerned with paternal certainty, often respond more strongly to sexual infidelity cues, whereas women, attuned to resource commitment, respond more strongly to emotional infidelity cues. Neuroimaging studies confirm this distinction, showing differential activation patterns in the amygdala, insula, and prefrontal cortex when processing these threats. Yet, biology alone does not dictate outcomes. Attachment style strongly mediates responses to perceived threats. Securely attached individuals tend to experience jealousy as a manageable emotion, using cognitive appraisal and communication to resolve conflict. Anxiously attached individuals, however, are prone to intense emotional reactions, rumination, and hypervigilance, which can paradoxically increase relational tension. Avoidantly attached individuals may respond with withdrawal, emotional distancing, or suppression, often undermining intimacy and leaving conflicts unresolved. The neurochemical basis of jealousy and infidelity is equally revealing. Dopamine and vasopressin, both involved in reward, pair bonding, and territorial behavior, heighten emotional responses to potential threats. Elevated cortisol during perceived infidelity amplifies anxiety and vigilance, sometimes triggering disproportionate behavioral reactions. Understanding these mechanisms allows couples to anticipate emotional escalations and implement strategies that regulate arousal, maintain perspective, and preserve trust. Preventing infidelity is not merely about policing behavior. It involves reinforcing relational bonds, sustaining desire, and maintaining emotional attunement. Regular shared experiences that generate novelty, pleasure, and achievement activate dopaminergic reward systems, keeping relationships dynamic. Rituals of appreciation, affectionate touch, and verbal affirmation continuously stimulate oxytocin, deepening trust and mitigating the allure of external alternatives. Subconscious processes also govern mate selection and fidelity. Humans communicate and evaluate compatibility through cues often outside conscious awareness. Pheromones, vocal tonality, body language, and micro-expressions. These signals convey genetic fitness, emotional state, and relational intent. Even subtle variations in scent can influence partner preference and sexual attraction. Couples who cultivate awareness of these unconscious cues often navigate conflicts and maintain sexual chemistry more effectively. Maintaining long-term passion requires deliberate engagement with both novelty and familiarity. While routine can stabilize relationships, it often diminishes dopamine-driven arousal. Introducing periodic challenges travel, learning new skills together, or cooperative problem-solving reactivates reward circuitry, sustaining excitement. Simultaneously, familiarity reinforces emotional safety and attachment, engaging oxytocin pathways that anchor desire to trust and intimacy. The interplay between novelty and stability forms a neurochemical rhythm essential for enduring desire. Jealousy, when properly managed, can also serve a functional role. Moderate jealousy signals investment in the relationship and motivates protective behaviors without overwhelming emotional regulation. However, chronic or excessive jealousy activates stress systems, erodes trust, and diminishes sexual desire. Couples can mitigate maladaptive jealousy through transparent communication, self-reflective awareness, and structured reassurance that reinforces commitment and security. Advanced strategies for maintaining desire include sensory priming and emotional attunement. Couples who regularly engage in eye contact, synchronized breathing, tactile intimacy, and emotionally expressive dialogue stimulate parasympathetic balance, oxytocin release, and reward sensitivity. These practices counteract the physiological habituation that naturally occurs in long-term relationships, ensuring that desire remains responsive rather than dulled. Neuroendocrine factors remain central to sustained passion. Testosterone fluctuations in men and women influence sexual motivation, pursuit behaviors, and responsiveness to novelty. Estrogen and progesterone cycles in women modulate emotional receptivity, sexual interest, and reward sensitivity. Awareness of these rhythms allows couples to optimize intimacy, timing encounters to align with peak desire periods. Supplements and lifestyle interventions such as exercise, stress management, and sleep regulations support hormonal balance and reinforce neurochemical readiness for sexual engagement. Subconscious mate preferences continue to shape long-term relational dynamics. Traits such as symmetry, genetic diversity, emotional expressiveness, and social intelligence influence sexual attraction and attachment security. Even after conscious commitment, these subconscious evaluations subtly influence desire, fidelity, and relational satisfaction. Partners who acknowledge and respect these influences often navigate challenges more effectively, fostering a balance between conscious choice and innate preference. Finally, love, desire, and attachment are adaptive systems, shaped by continuous interaction between biology, psychology, and behavior. Relationships that thrive over decades are those in which partners actively engage these systems, regulating emotional responses, reinforcing reward pathways, integrating novelty with stability, and consciously managing jealousy and fidelity challenges. Long-term intimacy is less about luck and more about intentional neurochemical, hormonal, and behavioral orchestration. In conclusion, the science of infidelity, jealousy, and enduring passion reveals that human relationships are both fragile and resilient. By understanding the underlying neurobiology, subconscious cues, and behavioral strategies, couples can transform potential vulnerabilities into sources of strength, deepening desire, reinforcing attachment, and sustaining the remarkable experience of love over a lifetime. As we continue our journey through the science of love, desire, and attachment, we arrive at a critical question. How did romantic love evolve, and how does it differ across the vast spectrum of human cultures? Understanding the evolutionary roots of love not only illuminates our behaviors, but also reveals the biological and psychological mechanisms that sustain long-term partnerships. Romantic love, in its essence, is a biologically adaptive system, designed to maximize reproductive success and ensure the survival of offspring. Neuroimaging studies reveal that romantic attachment activates brain regions associated with reward, motivation, and goal-directed behavior, the ventral tegmental area, the caudate nucleus, and the orbitofrontal cortex. These areas overlap with those engaged during intense pleasure, reinforcing the idea that love is a deeply motivating, neurochemically driven experience across cultures. The expression of love is astonishingly diverse, yet the underlying neurobiology remains remarkably consistent. In some societies, arranged marriages prevail, and romantic passion develops gradually over time. In others, courtship is driven by immediate attraction with intense early passion. Yet, despite these cultural differences, the same neurotransmitters dopamine, oxytocin, vasopressin, and serotonin mediate the feelings of attachment, desire, and trust. This suggests that, while cultural context shapes behavior, biology provides the scaffolding for emotional experience. Cross-cultural studies also reveal that mate preferences are universal in some respects, but flexible in others. Traits such as kindness, intelligence, emotional stability, and physical health are valued globally, indicating that certain qualities reliably predict long-term relational success. However, the weighting of these traits varies with local, ecological, social, and economic conditions. For example, in resource-scarce environments, indicators of reliability and resource provision may be prioritized, whereas in affluent societies, novelty, creativity, and social status may carry greater influence. Neuroplasticity, the brain's ability to adapt and rewire, itself plays a pivotal role in sustaining long-term attachment. Romantic relationships are dynamic, and the brain's circuits are continuously shaped by experience. Positive interactions, shared challenges, and mutual support enhanced connectivity in networks associated with reward, empathy, and emotional regulation. Conversely, chronic conflict, neglect, or betrayal can weaken these circuits, leading to reduced desire, diminished attachment, and heightened stress responses. Studies of long-term couples reveal that sustained passion is not merely a product of initial chemistry. Instead, it is maintained through intentional neuroplastic engagement, engaging in novel activities together, learning new skills, and emotionally attuning to each other's changing needs. These behaviors stimulate dopamine pathways, reinforce oxytocin-mediated bonding, and create a resilient network of attachment that can endure the inevitable challenges of life. Cultural rituals 2. Influence neuroplasticity and attachment. Shared rituals, whether daily greetings, symbolic ceremonies, or communal celebrations, act as repeated cues that strengthen neural pathways associated with partnership. These rituals not only signal commitment, but also provide predictable, emotionally rich experiences that reinforce attachment and satisfaction. That the evolution of romantic love is closely linked to pair bonding, a phenomenon observed in humans and a few other species. Pair bonding serves both reproductive and survival purposes. It facilitates cooperative parenting, resource sharing, and mutual protection. Neurochemicals such as oxytocin and vasopressin promote bonding by reinforcing positive feedback loops between partners. For example, affectionate touch, emotional support, and coordinated activity trigger oxytocin release, deepening trust and attachment, while shared challenges amplify vasopressin signaling, increasing protective behaviors and loyalty. Long-term attachment also involves the regulation of desire and novelty. The brain's reward system is sensitive to both familiarity and surprise. Stability and predictability provide emotional security and trust, engaging parasympathetic pathways and oxytocin-mediated bonding. Novelty, on the other hand, activates dopamine circuits, sustaining arousal and interest. Couples who skillfully integrate these elements maintain both routine and novelty achieve an enduring balance between safety and excitement, which is crucial for sustained romantic desire. Interestingly, neuroplasticity extends to the unconscious domain. Subtle cues such as pheromones, vocal intonation, facial expressions, and micro-movements shape attraction and attachment without conscious awareness. Over time, partners learn to predict, anticipate, and respond to each other's signals, enhancing emotional synchronization and relational efficiency. This subconscious attunement strengthens the neural architecture supporting long-term attachment, reinforcing both desire and trust. Another dimension is the role of adversity and challenge in long-term attachment. Stressful experiences, when navigated successfully, can enhance intimacy and reinforce neural pathways associated with partnership. Shared problem-solving, conflict resolution, and cooperative goal achievement create neurochemical and psychological reinforcement, increasing the couple's resilience and relational satisfaction. In contrast, unresolved conflict or chronic stress can erode neural connectivity in reward and attachment circuits, diminishing desire and trust. Finally, the evolution of love and attachment is intertwined with the broader social context. Humans are inherently social creatures, and our romantic decisions are influenced by kin, peers, and social norms. Social support networks amplify neurochemical reward systems, buffer stress, and reinforce attachment behaviors. Communities that value cooperation, shared rituals, and relational stability create environments that enhance long-term attachment and emotional well-being. In conclusion, the evolution and neuroplasticity of romantic love reveal that relationships are simultaneously biological, psychological, and cultural phenomena. Love is an adaptive system designed to foster long-term attachment, sustained desire, and mutual investment. Across cultures, the same underlying neural circuits and neurochemicals orchestrate our experience, while cultural practices and personal behaviors shape the trajectory of desire and attachment. Understanding these mechanisms allows us to intentionally cultivate passion, deepen attachment, and navigate the complexities of human love in ways that are both scientifically grounded and emotionally fulfilling. As we progress in our exploration of love, desire, and attachment, we now turn to a dimension that is both invisible and powerful: the hormonal and chemical signals that shape attraction, desire, and long-term partnership. While conscious choices and cultural norms guide much of our behavior, it is the subtle neurochemical and hormonal undercurrents that often determine who we find attractive and how desire unfolds over time. Hormones act as messengers between our internal state and the external world, influencing both perception and behavior. Among these, testosterone, estrogen, progesterone, and cortisol play pivotal roles in shaping sexual desire, emotional bonding, and relational decision-making. Testosterone, often associated with libido and assertiveness, operates not merely as a trigger for sexual pursuit, but as a modulator of confidence, social dominance, and mate selection. Estrogen, in both men and women, modulates sensitivity to attraction cues, enhances receptivity to sexual stimuli, and interacts with dopamine systems to influence motivation and reward. The interplay between these hormones is dynamic and cyclical. In women, the menstrual cycle creates fluctuations in estrogen and progesterone that subtly alter preferences, sensitivity to attraction, cues, and even olfactory perception of potential partners. Research shows that women may unconsciously perceive certain traits, such as symmetry, genetic compatibility, or social dominances, more attractive during peak fertility phases. Men, in response, may exhibit variations in testosterone that correlate with competitive behaviors and mate-seeking tendencies. Reflecting an intricate dance of biological synchronization, subconscious attraction operates through multiple channels, often beyond our conscious awareness. Pheromones, for example, are chemical signals that convey genetic compatibility, health, and reproductive fitness. Studies demonstrate that humans can detect and respond to these olfactory cues even without conscious recognition, shaping attraction, sexual desire, and partner choice. Beyond smell, subtle cues such as vocal tone, micro-expressions, posture, and gait also influence attraction, interacting with the brain's reward and limbic systems to reinforce desire and attachment. Long-term desire, however, is not merely a product of initial attraction or chemistry. It is sustained through both biological feedback and intentional behavior. The neurotransmitters dopamine, oxytocin, and serotonin coordinated experiences of pleasure, bonding, and emotional regulation, creating circuits that can be strengthened through shared experiences, novelty, and mutual responsiveness. For example, engaging in new activities together stimulates dopamine, reinforcing curiosity and excitement. Acts of physical intimacy and emotional support trigger oxytocin, deepening trust and attachment. Even small daily gestures, expressing appreciation, maintaining eye contact, or verbal affirmations enhance these neurochemical systems. Sustaining desire and commitment over time, the autonomic nervous system also plays a critical role. Sexual arousal and romantic desire involve a delicate balance between the sympathetic and parasympathetic branches. Sympathetic activation fuels anticipation and desire, while parasympathetic engagement is necessary for physiological readiness and emotional attunement during intimacy. Chronic stress, overactivation of the sympathetic system, or dysregulation of these pathways can inhibit desire even in the presence of strong emotional attachment. Conversely, practices that enhance parasympathetic tone such as mindful breathing, shared relaxation, and safe emotional environments support both sexual function and relational satisfaction. External factors, from lifestyle to environmental cues, further influence desire. Exercise, sleep quality, diet, and social interactions all modulate hormone levels, neurochemical balance, and autonomic function. Adequate sleep, for instance, maintains testosterone and estrogen at optimal levels, while chronic sleep deprivation can suppress libido and increase irritability. Similarly, physical activity increases blood flow, boosts dopamine, and enhances confidence and mood, creating fertile ground for desire and attraction. Over-the-counter supplements can modulate these biological systems to support libido and desire, although their effects are variable. Maca root, tonkat ali, and Tribulus terrestris have shown evidence of enhancing sexual desire in men and women through pathways both independent of and interacting with testosterone and estrogen. These substances highlight the diversity of mechanisms through which desire can be influenced: chemical, hormonal, and neurochemical. Yet, it is critical to remember that these are tools, not guarantees. Human desire emerges from an intricate web of biology, psychology, and social context. The mind-brain connection is equally powerful. Conscious anticipation, imagination, and emotional memory reinforce neurochemical circuits associated with desire. Thinking about a partner, recalling shared experiences, or anticipating future intimacy can release dopamine and oxytocin, enhancing desire even in the absence of immediate physical cues. Over time, these mental rehearsals contribute to the plasticity of attachment circuits, helping couples maintain excitement and engagement across the lifespan. Interestingly, desire and attraction are also shaped by the perception of scarcity and availability. Studies show that when individuals perceive their partner as responsive, attentive, and engaged, attraction to alternative partners decreases. Conversely, when responsiveness is inconsistent, the brain interprets this as a potential gap in reward, increasing sensitivity to novel or alternative options. This phenomenon underscores the importance of self-expansion and emotional responsiveness, concepts explored in previous chapters in sustaining long-term desire. Finally, maintaining long-term romantic desire requires intentional integration of novelty, emotional attunement, and biological awareness. Couples who cultivate curiosity, engage in shared challenges, and maintain physical and emotional intimacy activate dopamine, oxytocin, and serotonin circuits in coordinated ways. Hormonal awareness. Hormonal awareness is understanding how cycles, stress, and lifestyle affect libido provides a toolkit for aligning biological readiness with emotional and relational engagement. In summary, the science of long-term desire is a symphony of hormones, neurotransmitters, subconscious cues, and intentional behaviors. Desire is neither fixed nor purely instinctual. It is an emergent property of biology, mind, and social interaction, dynamically influenced by both conscious and unconscious processes. By understanding these mechanisms, we gain insight into sustaining passion, deepening attachment, and navigating the rich landscape of human love, from the sparks of initial attraction to the enduring bonds that define our most intimate partnerships. As we venture further into the intricate landscape of love and attachment, we arrive at a domain that is both intensely personal and profoundly biological: the neuroscience of jealousy, trust, and fidelity. These elements form the foundation of romantic relationships, shaping the boundaries of desire, security, and emotional investment. Jealousy, often perceived as a negative emotion, is in fact a deeply adaptive mechanism. Rooted in evolutionary pressures, it evolved to protect valuable bonds and ensure reproductive success. Neuroimaging studies reveal that jealousy activates regions such as the amygdala, anterior cingulate cortex, and insular areas responsible for threat detection, emotional processing, and social cognition. This neural activity reflects the brain's interpretation of potential loss or betrothal, signal that a vital social bond may be at risk. Interestingly, jealousy manifests differently in men and women, influenced by both evolutionary imperatives and cultural context. Men often exhibit heightened sensitivity to sexual infidelity, a pattern linked to the need to ensure paternity certainty. Women, on the other hand, may experience stronger emotional jealousy, reflecting evolutionary pressures to secure long-term resources and commitment for offspring. These tendencies, however, are not absolute. Individual variation and conscious regulation play substantial roles in moderating jealous responses. Trust, the counterbalance to jealousy, is equally crucial in sustaining relationships. Trust is mediated by the brain's oxytocinergic system, primarily involving the hypothalamus and limbic structures. Oxytocin, often called the bonding hormone, promotes social attunement, empathy, and cooperative behavior. When trust is present, oxytocin levels rise, enhancing feelings of safety, emotional openness, and attachment. Conversely, breaches of trust trigger cortisol release, activating stress circuits that can erode intimacy and provoke defensive or avoidant behaviors. Fidelity is a complex interplay between desire, trust, and self-regulation. The prefrontal cortex, responsible for executive function and impulse control, works in tandem with the limbic system to navigate the tension between immediate reward and long-term relational goals. Individuals who can engage prefrontal control effectively are better able to align behavior with values and commitments, demonstrating resilience in the face of temptation. Neurochemical modulators such as serotonin also influence fidelity, regulating mood, impulse control, and social behavior in ways that reinforce commitment. The dopamine system, central to reward processing, plays a paradoxical role in fidelity. Novelty and excitement trigger dopamine release, enhancing motivation and attention. Within a relationship, experiences of novelty, shared adventures, new challenges, and novel forms of intimacy can sustain dopamine activity, keeping desire and engagement alive. However, the same system can amplify the allure of alternative partners, creating a biological tension between exploration and attachment. Understanding this duality allows us to navigate desire intelligently, fostering both excitement and loyalty. Social context further shapes how jealousy, trust, and fidelity are experienced. Attachment style formed early in life through caregiver interactions. The intensity and expression of these emotions, securely attached individuals are more likely to exhibit trust, resilience to jealousy, and consistent fidelity. Anxious or avoidant attachment patterns can amplify fear, suspicion, or disengagement, demonstrating the lasting imprint of early neurobiological development on adult relationships. Cultural norms and societal expectations overlay these biological substrates, influencing perceptions of acceptable behavior, strategies for conflict resolution, and definitions of commitment. Cross-cultural studies reveal variation in jealousy triggers, tolerance for infidelity, and expressions of trust, highlighting that while biology sets the stage, culture scripts the narrative. Conflict and resolution mechanisms are central to sustaining long-term bonds. The brain's mirror neuron system, responsible for empathy and social understanding, enables partners to anticipate and respond to each other's emotional states. Constructive communication active listening, and emotional attunement stimulate oxytocin release and reinforce neural circuits associated with bonding. In contrast, chronic conflict or perceived betrayal activates stress pathways, releasing cortisol and potentially undermining attachment. Neuroscience also illuminates the pathways through which forgiveness and reconciliation restore trust. Research indicates that deliberate acts of empathy, acknowledgement of wrongdoing, and reparative behaviors can reset neural circuits, reducing amygdala hyperactivity and enhancing prefrontal regulation. This neuroplasticity demonstrates that even after breaches of trust, relationships can recover, provided the brain is guided by intention, empathy, and sustained effort. Sexual fidelity, while a prominent focus, is intertwined with emotional fidelity. Studies of romantic and sexual behavior suggest that emotional intimacy, shared vulnerability, support, and reciprocal understanding serves as a predictor of overall fidelity, often more powerful than sexual attraction alone. The neural mechanisms underpinning this include coordinated activity in the medial prefrontal cortex, orbitofrontal cortex, and limbic system, reinforcing attachment through rewarding emotional interactions. Technology. Modern dating and social media introduce new challenges to fidelity and trust. The immediacy of connection, visibility of alternatives, and curated presentation of self can intensify jealousy and temptation. The brain, evolved for small-scale social networks, must now navigate vast social landscapes, balancing novelty-seeking drives with attachment systems that evolved in a more constrained environment. Awareness of these pressures, combined with conscious strategies for regulation and transparency, can mitigate the impact of digital complexities on relationship stability. Ultimately, the neuroscience of jealousy, trust, and fidelity reveals that love is both fragile and resilient. The same circuits that detect threats and trigger protective emotions can, when guided by awareness, intention, and empathy, reinforce commitment, deepen trust, and sustain long-term desire. Understanding these mechanisms equips individuals and couples to navigate the delicate balance between vulnerability and protection, passion and stability, exploration and attachment. In essence, our brains are wired for connection, yet constantly negotiating between self-interest and relational harmony. By observing these neural patterns, respecting biological impulses, and integrating conscious decision-making, we can cultivate relationships that are both emotionally fulfilling and biologically resonant, capable of withstanding the challenges of time, temptation, and circumstance. As we reach the final chapter of our journey through the science of love, desire, and attachment, we confront the ultimate question: how do we sustain deep, meaningful bonds across the span of a lifetime? The neuroscience and psychology of love provide not just explanations, but strategies for cultivating enduring relationships, revealing the delicate interplay between biology, behavior, and conscious intention. Long-term attachment is grounded in mutual care, emotional attunement, and shared purpose. Couples who thrive over decades often demonstrate a remarkable ability to navigate life's challenges together, balancing intimacy with autonomy. Neuroscientific research shows that these enduring bonds are reflected in consistent activity in the ventral striatum, orbitofrontal cortex, and oxytocin-mediated pathways, reinforcing reward, empathy, and commitment. Desire, often imagined as a fleeting spark, is in fact a dynamic system that evolves. Romantic passion may peak early in relationships, fueled by dopamine and norepinephrine, creating intense pleasure and arousal. Over time, this initial surge is often replaced by a deeper, more stable form of attachment, what psychologists call companionate love, characterized by oxytocin-driven bonding, emotional support, and long-term investment. Understanding this evolution allows partners to anticipate changes, nurture intimacy, and sustain desire beyond the initial stages of romance. Effective communication is a cornerstone of lifelong attachment. Neuroscience confirms that the prefrontal cortex and mirror neuron systems play pivotal roles in empathy, perspective-taking, and conflict resolution. Couples who engage in active listening validate each other's experiences and navigate disagreements with respect and curiosity, not only reduce stress, but also reinforce neural circuits associated with trust and emotional connection. Shared experiences, novelty, and joint goals stimulate the dopaminergic reward system, keeping desire alive and reinforcing the sense of partnership. Couples who prioritize adventure, creativity, and mutual growth demonstrate heightened neural activity in reward-related areas, suggesting that novelty and shared purpose are essential not only for excitement but for long-term attachment. Attachment styles continue to influence behavior across the lifespan. Securely attached individuals tend to experience lower conflict, greater emotional stability, and higher relationship satisfaction. Conversely, anxious or avoidant attachment patterns can resurface during periods of stress, highlighting the importance of self-awareness and conscious regulation. Couples who recognize these patterns and actively engage in adaptive strategies, mindfulness, reflective dialogue, and mutual reassurance can mitigate potential disruptions and deepen relational resilience. Trust remains a central pillar. Lifelong bonds are continuously reinforced through consistent, reliable behavior, transparency, and responsiveness. Neurochemically, trust involves oxytocin release, cortisol regulation, and prefrontal limbic coordination, demonstrating that emotional reliability is not abstract but a tangible neurological process. Breaches of trust, while challenging, can be repaired through intentional acts of empathy, communication, and restorative behaviors that reshape neural pathways and restore emotional equilibrium. Sexual intimacy, while evolving over time, remains integral to attachment. Research indicates that physical touch, affectionate gestures, and sexual connection reinforce oxytocin and dopamine activity, maintaining both desire and emotional closeness. Couples who maintain intimacy, even in subtle ways, strengthen bonds, enhance well-being, and sustain attachment into later stages of life. Forgiveness and adaptability are essential strategies. No relationship is immune to mistakes, misunderstandings, or conflicts. Neuroscience shows that practicing forgiveness reduces amygdala reactivity, calms stress circuits, and promotes prefrontal regulation, enabling couples to move beyond resentment and rebuild connection. Emotional resilience, cultivated through reflection, communication, and shared meaning, transforms challenges into opportunities for growth. The role of gratitude and appreciation is equally profound. Regularly acknowledging a partner's contributions, expressing admiration, and savoring shared experiences activates reward pathways and positive affect networks, fostering satisfaction and reinforcing long-term commitment. Small, consistent acts of recognition, whether verbal, physical, or symbolic, create cumulative neural and emotional effects that support enduring love. Life transitions, career changes, parenting, aging, or loss test the strength of attachment. Couples who navigate these transitions successfully demonstrate flexibility, shared problem-solving, and adaptive coping. Neuroscience suggests that coordinated stress regulation, joint goal setting, and emotional synchronization during these periods reinforce both relational stability and individual well-being. Finally, love and attachment are not merely biological phenomena. They are human practices cultivated through intention, reflection, and sustained effort. Awareness of our neural predispositions, dopamine-driven desire, oxytocin-mediated bonding, cortisol-reactive stress combined with conscious strategies for empathy, communication, and novelty, empowers us to create relationships that are not only enduring but profoundly fulfilling. In conclusion, the journey through desire, attachment, and fidelity reveals that love is simultaneously fragile and resilient, biological and behavioral, ephemeral and enduring. By understanding the science behind our emotions, respecting the complexity of our brains, and committing to conscious cultivation of intimacy, we can navigate the challenges of human connection. Lifelong love is not the result of chance, but of consistent, informed effort guided by both heart and mind, biology and intention. As we close this exploration, we recognize that love is, at its core, a practice, a continuous interplay of curiosity, care, and commitment. In understanding ourselves, our partners, and the intricate dance of attachment, we find the keys not only to sustaining relationships, but to unlocking the profound joy, resilience, and wonder that human connection can provide. And so, we arrive at the edge of our journey, a journey through the hidden landscapes of desire, love, and attachment. What we've explored is more than science. It is a mirror to ourselves, a reflection of the intricate ways our brains, bodies, and hearts are wired for connection. Love, as we've seen, is both simple and complex. It is the spark of attraction, the chemistry of desire, and the deep currents of lifelong attachment. It is shaped by molecules we cannot see, hormones we cannot feel, and neural pathways we are only beginning to understand. And yet, it is equally shaped by our choices, our attention, and the small, deliberate acts of care we perform every day. The science tells us that we are not passive players in our relationships. Our brains, our autonomic systems, and even the stories we tell ourselves about our partners all converge to shape how we see the world and how the world sees us. We learned that desire can be cultivated, that attachment can be strengthened, and that the stability of love is grounded in both biology and conscious intention. And here lies the wonder. Every interaction, every word, every shared experience is an opportunity to expand ourselves, to deepen connection, and to create bonds that are resilient, joyful, and enduring. The science gives us insight, but the true power is in how we apply, is how we choose to listen, to respond, to show up fully for the ones we care about. So as you step back from this exploration, remember this: love is not only felt, it is practiced. It is not only given, it is cultivated. And in understanding the beautiful complexity of our minds, our bodies, and our hearts, we unlock the ability not just to experience love, but to transform it into something extraordinary. This is the Wonder Lab. Thank you for joining us on this journey. Keep exploring, keep questioning, and never stop seeking the wonder in the world around you and within the hearts of those you hold dear.